own hand by mistake.
You mean like this? Or like this? Or this? Or this? But what about this? Or this? Or this? Or this?
Except he wasn't a sponge.
So?
What's up, everybody? Today we are talking DNA replication, so how to start off with one strand of DNA and end up with two. This process is called semi-conservative, meaning that we start off with a parent strand, and basically we take this parent strand and we split it in half, and we take each half and make a new strand of DNA. So let's take a look at how this works. So if we're talking about replication as a whole, it has a process that involves lots of enzymes, and the enzymes are like the workers that get the job done. First enzyme we're going to talk about is called primase. He tells us where to start. Helicase is what splits the helix. We have DNA polymerase. Since we were making a polymer, we use a polymerase to make this DNA polymer. It's going to add the letters to it, so it's going to add the new A, T, C's, and G's. We call them nitrogen bases. And then we have ligase, and I think of ligase as the glue. He glues it all back together for us. He actually synthesizes new sugars and phosphates for our backbone. So moving forward, if we look at our replication fork, it looks literally kind of like an old fork. We have our nitrogen bases that are bonded together by hydrogen bonds. And the first thing we need to do is we need to know where to start. Well, in order to know where to start, primase is like a flag, like we stuck on the moon, and he says, hey, you need to start here. And who he tells that to is a wedge, and this guy who is a wedge, his name is helicase. Remember, DNA is a double helix, so we need a helicase to break it. He is literally going to break the hydrogen bonds by a wedge, kind of like your zipper. There's that wedge that zips and unzips your jacket. So if we have all of our nitrogen bases here in our fork, we know that DNA is made up of different ends, 5' on one end, 3' on the other, and they're opposite on the other side of DNA. The first guy that's going to come in after it's split is called DNA polymerase. He's going to make a new polymer called DNA. He's going to read the strand that's already there, and he's going to pair up new nitrogenous bases with the existing parent strand. So he sees a C right here. When he sees that C, he's going to add a G, and so on and so forth. Now, he can go as fast as he wants to on this side because helicase is going to split nitrogenous bases in front of him. We say that this is the leading side or the leading strand. Now, the other side, though, he can only move from 5' to 3' on the daughter strand. Well, when he does this, he's going to start at the top and he's going to work his way down. So we call this the lagging strand. But here's what happens with the lagging strand. He started at that T and he worked his way all the way down, but then we had helicase kept working and kept splitting new bases. So he worked his way down that strand. Now he's got to come back up to the top and start again. When we do this, we're going to end up with a gap. See how I have my C and my T right here? Well, he already made the A, T, C, G on the other side, so we end up with something called an Okazaki fragments. For that reason, we call this the lagging strand. Think of your internet as lagging. It's going to have different segments in your bandwidth. So what do we need? We need somebody to come in to attach all these segments together, and his name is ligase. He's going to go through, he's going to connect the Okazaki fragments, again, named after the guy who discovered them, and he's going to build a sugar phosphate backbone for everybody. His name is ligase. He's like the glue that holds everything together. All right. So let's take a recap because there's a lot of information here. We have a strand of DNA. We call it our parent strand. We're going to take our parent strand. We're going to put a little flag on it. We're going to say, hey, we need to copy the hair color gene, so we need to start right here. Helicase is going to break the helix. It's going to unwind and unzip it. Once you do that, we're going to have a DNA that's split literally on either side. There's no more hydrogen bonds in between them. They've been broken, as you can see. So we need somebody to come in and copy the existing nitrogenous bases there. So we have two copies. That guy's name is called DNA polymerase. Again, DNA is a polymer. We need a polymerase to make a new polymer. He's going to add the new letters, aka the nitrogenous bases, A, T, C, and G. Once he does that, the only thing left to do now is to glue them back together. So our guy that's the glue, he's going to come in. We've got our nitrogenous bases here. His name is ligase once again. We need those sides of this ladder to hold it together. So ligase is going to come in and he's going to synthesize new sugar phosphate backbones for the red letters there, the daughter strands. So my little star here that I'm so good at drawing at is going to swoop in. When he goes down that line, it's going to form a new backbone, making a new strand of DNA. Again, he builds the backbone, the sugar phosphate backbone. Very important. Now, this process is semi-conservative, meaning I did not start off with one white strand and make one green strand and one white strand. That's not how we do it. I start off with a parent strand that splits in half, and now I have half parent and half daughter in the new sides. Two different colors because they came from half the parent and half the daughter. It's not a complete copy. We split it, we build it back from one half. And that is DNA replication.
So it didn't grow back!